All right, so welcome to the is it May? It's April edition of Micro Seminar. Today is, it's almost May. The semester's almost over, so I'm a little bit wonky in terms of timing. But today's Micro Seminar, we're gonna actually delve into the world of plant microbes and plant pathogenicity. And so today's speaker is Dr. Brian Kvitko. I've known Brian for a while. I will say this, I teach microbial genetics, and Brian's the guy I go to for microbial genetics questions, because he's pretty spectacular in terms of just manipulating genomes and understanding how things work. So Brian started as a grad student in Alan Colmer's lab in Cornell, working with Pseudomonas syringae, and then took a postdoc in Colorado State with Herbert Schweitzer, actually working on Burkholderia. So the challenge of Burkholderia is designing, because a lot of the strains are select agents, the challenge is actually designing markers to manipulate the genome with, because the government doesn't like Burkholderia to be resistant to things. So he was working on that for a little bit, designing ways to manipulate Burkholderia genomes, and then went back for another postdoc in Pseudomonas syringae with Shengyang He. And after that, started his lab at the University of Georgia about 2 years ago. Today, he's actually coming to you live from southern Georgia, where he's working on a field experiment that I was supposed to participate in, but then dropped the ball and couldn't actually make the mutants, but he's doing it nonetheless. So without further ado, Brian Kvitko.
Okay, thanks, Dave. I appreciate it. All right, I'm going to, let me do my screen share, and I will get into my slides. Thanks, everybody, for joining me today. Okay, so the title of my talk, as I guess you guys probably know from Twitter already, hold on, let me switch over, is Dazed and Confused. How does plant immunity suppress bacterial virulence? Which is the big question, and the big question that we've kind of, that I'm trying to structure my program around. As Dave mentioned, I used to work in infectious disease, working on select agent human pathogens. So I had a lot of colleagues, and I would tell them about, you know, when we used to work with plant pathogenic bacteria, and I don't really think they got it. They didn't understand sort of why would you study the plant immune system. So my sort of 30-second elevator pitch version of this was this, that almost any plant can stop the growth of almost any microbe. That's not just plant pathogens, that's any microbe. And we don't really know how they do it. So hopefully that'll catch your attention. Okay, so first of all, a lot of people don't really realize this, and maybe some people do. Plants can get sick. They can get sick in the same way that any other multicellular eukaryote can. And there's a wide variety of organisms that cause plant disease.
Hey, Brian?
Yeah.
Heads up, you got a little tab saying you're sharing your screen on the bottom, so I think there's a clicky thing to just make it go away. So
yeah, is that better?
That is better.
Okay, so far and away the most common plant pathogens are fungi, but there are all different classes of plant pathogens, fungi, oomycetes, viruses, nematodes, as well as bacteria. Now bacteria obviously offer some key advantages for study. They replicate rapidly as asexual clones. It's very simple to culture, typically, not all the time, but typically. They have small, compact, and, you know, quote-unquote haploid genomes, which is always nice. You don't have to worry about 2 copies of everything getting in your way. And far and away, they're amenable to very precise genetic modification. So the particular system that we work on, or that we use, an excellent sort of model pathogen is the bacterial pathogen Pseudomonas syringae. It's a gram-negative plant pathogenic bacteria, and strains collectively infect a wide variety of hosts, you know, from dicots, monocots, and woody species, causing a huge variety of symptoms. Spots and specks, blights, and here we have bleeding canker on kiwi, very dramatic. And individual strains are typically host-specific and divided roughly into pathivores, usually on their host of isolation rather than any stringent assessment about their total host range. The pathogenic life cycle of Pseudomonas syringae is that it typically infects aerial tissues, gains access to the leaf interior through wounds or natural openings such as the stomata. See here the little air ports that are used on leaves for gas exchange. And a key thing to remember is, unlike a lot of animal pathogens, In plant pathogens, the bacteria will actually remain outside of the plant cell wall, and they replicate in the intercellular spaces within those leaves, as opposed to being internalized into the cells themselves. Okay. So the particular organism that I work on is Pseudomonas syringae pathovar tomato DC3000, which I will just call as DC3000 because the rest of it is a bit of a mouthful. And it really has been an excellent model host to look at and study plant disease and plant immunity. Not the least of which reason because it can infect Arabidopsis thaliana as an alternative host. So you have an excellent, annotated genome, a very tractable organism on both your pathogen side and your host side. It also infects the horticultural crop tomato, and it can infect an alternative host, Nicotiana benthamiana. So if Arabidopsis is sort of the white mouse of the plant world, I would say benthamiana is the white rat of the plant world. Let's see. So how does Pseudomonas syringae cause disease? What are its virulence factors? So one key thing for the virulence factors in Pseudomonas syringae is that for the most part, almost all of the virulence factors that are known are regulated using a HARP-L virulence promoter, or a HARP box, which has this consensus motif you can see here on the screen. HARP standing for hypersensitive response and pathogenicity. Basically, these are the genes that are required for it to interact with plants in a meaningful way, either to cause disease or to activate a defense response. The genes in that regulon are tightly regulated with one another. Here you can see this is a time course done in vitro by microarray. Now, what is in the HARP-L virulence regulon? Well, one thing that you have is caronitin. So caronitin is a non-host specific toxin that mimics a plant hormone, jasminial isoleucine, which actually has the smell, well, actually, I guess it's methyl jasmonate, actually gives jasmine flowers their unique odor. It's actually a defensive compound. And the pathogens use that to sort of trick the plant into defending itself in the wrong way. They can also use it to cause stomata to reopen, allowing them to invade into the leaf. And then, in addition to that, in the HARP-L virulence regulon, we have the type 3 secretion system, which is basically a proteinaceous needle-like structure, which it uses to bridge across both the inner and outer membrane of the bacteria, cross the plant cell wall and the plant host membrane to deliver proteinaceous effectors directly from the bacteriocytosol into the plant host cell. The use of a type 3 secretion system is actually a common virulence strategy used in many bacterial plant pathogens of both animals and plants. Okay, so the type 3 secretion system is actually, it's extremely important for the infection of Pseudomonas syringae to the point where if you were to disrupt this structure, it's essential. If you do not have a functional type 3 secretion system, you do not see disease caused by Pseudomonas syringae on plant hosts. So why is that? Why is this type 3 secretion system such an important virulence factor? Well, to get a good handle on that, I actually want to talk a little bit about how plants defend themselves. So how do plants protect themselves from pathogens? Okay, so plants get sick. Well, another thing that's important to realize is that plants also possess an innate immune system. In fact, we usually think of the immune system as being antibodies, the adaptive immune system. But the truth is that the vast majority of multicellular eukaryotes on this planet get by perfectly fine with just an innate immune system. And adaptive immunity is pretty much restricted to, a subset of vertebrates. So let's see. I'm sorry for the 5 kingdom model, by the way. That's the best figure I could find of this. So how does this work? So one of the key sets of receptors that are used in both plants and animals, one of the key mechanisms by which they will detect the presence of pathogens is by monitoring for these pathogen, or more accurately, microbe-associated molecular patterns. So a great example of this is the detection of the bacterial flagellum. So here we have an image of the bacterial flagellum monomer. You can see what we have here if you look at this filament structure. The outside is actually incredibly variable. The inside core of the flagellum is actually highly conserved, except for these 2 little patches here where you see a high level of conservation, but it's more variable than the rest of that. Now, both animals and plants have leucine-rich receptor type proteins that span the membrane, and then have signaling domains in the cytosolic side of the membrane that can detect different motifs found on flagellin. When they detect those motifs, they then undergo a signaling cascade that leads to the activation of a defense response. And the animal version, the TLR5, actually detects a different region of flagellin than FLS2, which is the plant version that detects the same protein. Okay. All right. So this type of immunity, this detection of pathogen-associated molecular patterns, in plants, we call that pattern-triggered immunity or PAMP-triggered immunity. And this is the first line of defense in plant immune detection. So understanding how this works kind of gives some perspective on why the type 3 secretion system is so important. Okay, so let me show you a simplified model of how this is supposed to work. So we have our receptor-like kinase, which in this case is FLS2. Now, when a bacteria is present, it can detect those non-self molecular patterns, things like bacterial flagellin. It can detect peptidic glycan on fungi. It can detect chitin, all of these sort of conserved features that can't be easily lost. So upon that detection, you get the production of a signal cascade that leads to the production of a defense response. Now, in an adapted pathogen like Pseudomonas syringae, they will produce a type 3 secretion system and deliver effectors into the plant cytosol, which then spread out and target various plant targets, modifying those plant targets in such a way that this signal becomes dampened. This leads to a decrease in the defense response and creates a state of susceptibility, allowing the bacteria to cause disease. So one key thing to think about is that immune signaling and pathogen signal dampening are typically occurring simultaneously. So they're actually in a race with one another. And successful pathogens are able to dampen immunity rapidly enough that they allow disease to progress. Okay. But what if you give the host a head start? Okay, on the left side of the screen, what we have here is a crystal structure of the FLS2 receptor-like kinase with its co-receptor bound to an epitope of flagellin. This is the minimal portion of flagellin that the plant FLS2 receptor can recognize. So if we take just that peptide, that 22 amino acid peptide, and we pre-inoculate a leaf with that peptide, we can actually pre-induce this type of immunity. We can turn pattern-triggered immunity on ahead of time. So if we do that, if we pre-treat the leaves for 20 hours with 500 nanomolar of Flag22 and then infiltrate with the bacteria, you can see that the change in behavior is quite dramatic. The infection of these leaves by the pathogen, you can see that they're completely annihilated. Meanwhile, after 4 days, 4 days later, on the other hand, the ones that were pre-treated with Flag22 look completely green, completely healthy. This pre-activated PTI is actually very potent. Some of the things that we see are that the delivery of type 3 effectors through that secretion system is basically completely blocked. Bacterial replication is suppressed, symptoms are reduced. The plant is basically fully protected from the bacteria, even though we're infiltrating with what would normally be a fully competent pathogen able to suppress the immune response. So this is the question. What conditions does P. syringae encounter during immunity that alters its behavior so drastically? How do you take a normally competent pathogen and shut it down and basically make it behave in this way? So we did a little bit of armchair biology and kind of sat down and tried to think about, well, what are some different hypothetical classes? What could we predict might be explaining this behavior? So I would say the classic model is probably one of antimicrobial. This idea that the plant cell, when it's induced, will actively deploy antimicrobial compounds, be those small molecular weight compounds, hydrolytic enzymes, antimicrobial peptides, and so on and so forth. Another idea that's come to the fore recently is this idea of nutritional immunity, where the plant cell is actually sequestering key nutrients. So remember, the bacteria are occupying this intercellular space between the cells. So if the cells are able to basically starve them out by sucking up essential nutrients from that space, then the bacteria presumably wouldn't be able to grow. We have this idea of counter-regulatory immunity. the pathogens must be able to activate their their virulence functions in an appropriate way, at an appropriate time. They only turn them on when they need them, when they're actually inside the plant, and they can tell that. Well, one approach that the plant could use would be simply not presenting those signals that the pathogen would use to activate its virulence genes. And the last is this idea of physical immunity, which is you block pathogen access in some way. The type 3 secretion system would be prevented in some way from getting through the cell wall and making a functional contact. Now, the truth is that these are helpful sort of categories. There's a good chance that, like, if these were true, they probably could easily fit into multiple categories, and most likely there are multiple categories at play. Okay. So what are the mechanisms of bacterial inhibition by plant immune outputs? Basically, what do bacteria experience during plant immunity? How are we going to find that out? So our primary hypothesis was that the restriction of bacterial colonization during plant immunity is associated with specific host-created biological stressors, oxidative damage, the release of antimicrobial compounds. That was the hypothesis that we were seeking to test. So how were we going to figure out what those actual stressors were? Well, the approach that we thought was that the bacteria actually have a front row seat to immune defenses. If you really want to get a sense about what the efficacious responses are of immunity, best to look at the organisms that are being subjected to them. So the approach that we chose to take was to look at the D3000 transcriptional response to infer the stressor environment during that pre-activated immune state in plant tissue. Okay. So how did we do this? So we isolated total RNA from inoculated leaves. So we pre-induced with Flag22, or we had naive plants, we put bacteria in there, and then at certain time points, we just took the leaves and we ground them up, got all the RNA out. All right? So the problem is that the bacterial RNA is only there in a very small percentage. So what do we need to do? We need to get rid of everything else. So we need to deplete the plant cytosolic and organella ribosomes. Ribosomes obviously make up a ton of RNA. We depleted the bacterial ribosomes. We wanted to do everything we could to get to just the bacterial transcripts. We depleted all the polyadenylated eukaryotic mRNAs. Unfortunately, bacteria don't have that sort of universal feature on their mRNA that we can use to pull them out. So we have to take this approach of getting rid of everything else to the best of our ability. So we did all of this. We did all of the purification that we could think of. We submitted it for sequencing, and it was a complete failure. Basically, we got about 1 to 2% of bacterial reads back from this assay. And in retrospect, it was really obvious that this is what we would have seen, and we didn't really think about it at the time. But chloroplast transcripts, chloroplasts basically, for all intents and purposes, are bacteria, or at least their transcripts certainly look like bacteria. So what we ended up happening is we just kept sequencing the same photosystem transcripts over and over and over and over again, and pretty much it killed our read depth. So total RNA didn't really seem like it was going to work. So what was another approach? So this is what we sort of started working on and what we came up with when I started my program at UGA. So instead of trying to sequence total RNA, we chose to try to use a physical separation of the bacteria from the tissue. So we started like this. We infiltrated our Rhabdopsis with a high dose of bacteria, about 30 plants. Okay. At the desired time point, we removed those leaves and infiltrated the leaves with an RNA stabilizing buffer, kind of a homemade version of an RNA later. The idea there is that it would actually sort of freeze the bacterial transcriptome in place in whatever their in situ conditions had been. Then we use low-speed centrifugation to spin those bacteria out of the leaves where we can collect them by filtration. At that point, we did normal RNA prep, and we still did bacterial ribosome depletion. We did an RNA-seq time course to compare the bacterial transcriptome of D3000 in both naive tissue and pre-induced immune tissue at 1, 3, and 5 hours post-inoculation. And then we submitted that for library prep and sequencing. So we saw vast improvements in our read counts from this. We saw everywhere from 12 to 46% just pathogen. Now we do still see plant RNA from here, but this is giving us enough that we can actually get the kind of read coverage that we want. So using QRT to kind of balance the amount of bacterial RNA in these mixed samples, we then submitted that for, I think we did next-seq analysis. Was it high-seq? I'll have to double-check my notes. Anyway, the real point is that in each of our samples, we were able to achieve at least 7 million reads matching DC3000. And that was our goal, to have between 5 and 10 million non-rRNA reads for all the samples. So now what are we going to do? We got all this great data. How are we going to analyze it? So we built a pretty basic, straightforward sequence analysis workflow, starting with quality control and trimming, then mapping against the well-annotated DC3000 genome. Those reads were normalized based on total read count, and then used DSeq to do differential expression analysis, did pathway mapping using KEGG, and the only thing we haven't quite gotten to yet is the meme analysis. We still want to do some co-regulation to kind of feed that in and see what sort of regulatory motifs are associated with those co-regulated genes. Okay. All right. So now I'm going to be brave. I'm going to talk about all unpublished data. But what did we find? Okay. All right. What are the mechanisms? We've got this great transcriptional data. So what is it telling us? What is the bacteria experiencing during plant immunity? What amazing lessons does it have to teach us? What did we not find? We did not find any evidence for differential exposure to classic biological stressors. Reactive oxygen, osmotic stress, all of these various stresses, were they there? Yes. Were they differential between disease and immunity? No, they were not. There is a very obvious signature of osmotic stress in bacteria that are inside of the plant tissue, but it's not differential. It's equal between both naive and immune-induced plants. So what did we find instead? Instead of antimicrobial effects, sort of these biological stressor effects, what we seemed to find instead were these counter-regulatory-like signatures. So we saw altered regulation of key virulence factors. So these include the carnitine synthesis genes, that's that non-host specific toxin I talked about, altered regulation of flagellin and chemotaxis genes, as well as, in particular, pretty significant alteration of the harp-L-regulon. Okay, so let me just break those down a little bit. So the genes for carnitine synthesis basically are only very weakly induced during immunity compared to during exposure, during disease. During disease, you can see that they're very sharply and very strongly upregulated by 5 hours, whereas under immunity, basically there's most of these values aren't even statistically significant. You can see we've got 2 operons here that go into the synthesis for the 2 different moieties, CMA and CFA, but pretty much every single one of those genes is very significantly downregulated during the immune response. So we also saw an interesting thing, that flagellar and chemotaxis genes are less strongly repressed during immunity. So basically during a disease, what we see is this very sharp and very quick and very strong repression of flagellar synthesis genes and chemotaxis genes. You can see here the darker blue colors are the ones that are associated with a reduced expression relative to sort of the mock media baseline control. Whereas under immune conditions, basically the gene expression of these flagellar genes is almost stable compared to the KB media that they started on. So we thought this was kind of interesting. First of all, I'm not sure if it was known that the flagellar system would be so quickly and dramatically downregulated during plant disease. But it does kind of make sense. The flagellin is one of the most, it's one of the things that plants really do a good job at detecting. So you would think that the pathogen would actually want to shut those down if it didn't need them to prevent immune detection, or at least limit it. So could this increased flagellar expression associated with immunity basically be creating sort of a feedback loop where it's associated with increased immune signaling by the plant? Okay. Now, another thing that we saw is that most of the type 3 associated genes are less strongly induced during immunity. You can see here the pink bars are the expression of the different genes under immune conditions at 3 hours. This is when we saw peak expression for both naive and mock conditions, naive and immune protected conditions, excuse me. And you can see that, yeah, they're induced. I mean, they definitely are relative to the immediate control, but they're always more weakly induced than during disease. In fact, there's about a 2 to 6-fold difference between the level of expression during disease versus immunity. And again, these are the structural components make up the needle itself. The helper proteins are thought to be associated with the translocon or degrading the peptidoglycan layer in between. And then these are the effectors. These are these protein cargoes that get delivered through the pilus into the plant host cell. Now, most of them were less strongly induced. A few of them seem to have sort of interesting conditions. Some of the effectors were actually conditional. So they weren't activated during immunity at all. They were only turned on during disease. And some of these have been shown to be quite important to plant disease, such as HOP-R and HOP-K1. These are effectors with known functions. And then another subset seemed to be delayed. There was no expression seen at 1 hour, for instance. And eventually, when they were expressed, they never even closely caught up to their diseased counterparts. Okay, so that's interesting. We're seeing all these basic virulence factors, their regulation being very, very different during immunity than during disease. Now, there was one more surprise kind of hiding for us in this data, and that was this. We see one of these type 3 substrates, one of these cargo proteins able to move through the type 3 secretion system, was actually only induced during immunity. Never saw expression of it, never saw differential expression of it at all during disease, but definitely turned on during immunity. So this is a member of the HOP-AH family. And what's interesting is this effector was basically ignored for a long time because we didn't think it was expressed, or it wasn't expressed during disease or any of the conditions that look like they would mimic disease. But it is a real substrate. This is a graph I won't go into now, but basically this tall bar indicates that it's actually able to pass through the type 3 secretion system and into the cell. But like I said, the HOP-AH2 family basically had been discounted, even though it's very common, very widely distributed in a large number of syringae strains. No member of the AH2 family has ever been seen to have one of these HARP-L dependent HARP box type promoters that are thought to be associated with virulence. Okay. So interestingly, HOP-AH is extremely broadly distributed in P. syringae. This is from Dave's paper in 2011. Let me zoom in here for a second. You can see these effectors are ordered from left to right based on their level of conservation. And you can see that HOP-AH is right at the top of the list. It's one of the most common type 3 substrates, but again, was basically completely discounted because we didn't see it being expressed at the same time as the type 3 secretion system. Okay, so is it actually, so I told you it's a cargo, it can pass through the system. As opposed to being an effector, is it actually one of these helper proteins, one of these things that are associated with aiding the process of delivery of other effectors? So D3000 actually carries 3 different HOP-AH family members, HOP-AH1, and then HOP-AH2, 1 and 2, 2, which are immediately adjacent to one another. None of these effectors were thought to be expressed, or there's no evidence for them being expressed. All 3 of them are legitimate substrates. They all can pass through the type 3 secretion system. And they're interesting in that they all carry a glycoside hydrolase family 1 domain. So what is that? So these are things like beta-glucosidases or beta-galactosidases. So this is the enzyme family that, for instance, includes cellulases. So I say that they could be a helper because they carry similar sort of domain structures to other type 3 helpers from plant pathogens. So these often will have a type 3 secretion system recognition target on their N-terminus, and then a plant cell wall or a bacterial cell wall-like domain on their C-terminus. So for instance, there are two proteins that are known helpers, HARP-W and HARP-AK, that carry pectate lyase-like domains. And then HOP-P carries a lytic transglycosylase domain that would interact with peptidoglycan. So similar to these proteins, the HOP-AH family carries, a cellulase-like domain. They're also cysteine lacking, which is a sort of odd thing that pretty much only this subgroup of type 3 substrates seem to have, is this lack of cysteine, although the meaning of that isn't really clear. So what does this mean? Why is this turned on? So there's a hypothesis that I've been floating and talking to Dave about a little bit. What if HOP-AH could aid in countering that established physical immunity? So during immunity, you'll get these depositions of a polysaccharide called callose, which is a little bit like cellulose, but with a beta-1,3 instead of a beta-1,4 linkage. And that gets deposited right against the cell wall and can actually help to block penetration by fungi. Well, what if HOP-AH is helping to basically degrade a structure like this and allowing the type 3 secretion system to then make a functional contact? Okay. All right. So summary and future work. So what did we find? Exposure of P. syringae to plant immunity dramatically reduces the expression of chromatin synthesis, blocks the repression of flagellin genes, and reduces the expression of type 3 associated genes 2 to 6 times. Interestingly, it induces the expression of the HOP-AH21 candidate helper gene. So what are we going to do next? We're going to try to use metabolomics to ID some of the candidate plant signals that affect these virulence gene regulations. So for instance, chromatin has a known two-component system that's the ultimate upstream regulator, but the signals that it perceives are not known. But now that we have this system, we might be able to identify metabolites that are key regulators of that virulence subsystem. We're also going to screen to see if we can figure out, what are the genetic regulators of HOP-AH21 and what's co-regulated with it. Maybe there's a whole series of these immunity-induced effectors. I know a lot of these other hypotheticals that we've seen pop up. Maybe these are also type 3 substrates that were never recognized previously. And then we want to determine if the HOP-AH family members contribute to countering this sort of established immunity. Okay, so with that, acknowledgements. I would like to acknowledge my lab members, Amelia Lovelace, my senior grad student. Most of the work that I've talked to her today is stuff that she pushed her way through and figured out the extraction symbol process and the analysis. Amy Smith, my technician, who's down here with me in Tifton, out in the field every morning collecting plant tissue. My new students, Sean Stice and Don Gillis, and our honorary lab members, Spencer Stumpf and Zach Mateen. My collaborators are Xinyang He and Andrea Velasquez, and we just started a new collaboration with Gail Preston to look at the metabolites. The work was funded by the University of Georgia and federal Hatch funds. And this is our lab challenge coin here with our lab logo that you get when you work in the Kvitko lab long enough to do some lab chores. And with that, I will happily take any questions. Okay. All right.
All right. Yeah, I'm back. No, I'm half changing a diaper, so that was my.
Do I have to cancel out my screen share, or am I already canceled out?
You can cancel out the screen share. That works.
How do I do
yeah, so my question is this. So what about the other helpers? So what's the difference between these helpers that they might, why would you have different helpers if they are all, are there different, this is actually something I don't know. Are there different, I guess there's different things they target or what?
So honestly, the helpers are kind of a weird situation. So one interesting thing about plant pathogenic bacteria as opposed to animal pathogenic bacteria and the secretion systems operate. Animal pathogenic bacteria have an absolutely conserved triad of proteins that form the translocon tip found at the end of the pilus that are responsible for forming that pore. So in plant pathogens, those proteins are completely absent. So what we seem to have instead is sort of a loose collaboration of proteins that seem to be able to perform partially one another's functions. So those are the harpins, which is, you know, kind of digging a bit deep, but the helpers are like that. So things like the HARP-W and HARP-AK, basically in the absence of any other known translocator, they seem to be able to facilitate some limited amount of delivery of proteins into plant cells. They work best as a group, but each of them can individually provide some activity. So those are the helpers that are involved at the translocon, sort of at the plant plasma membrane and the cell wall. The HARP-P and the HARP-AJ and the HARP-H, those all seem to be associated with the peptidoglycan. So it seems like those are responsible either for getting through the peptidoglycan in the first place, or I personally think it's possible that what they're doing is sequestering peptidoglycan fragments to prevent recognition by other receptor-like kinases, but that's an untested theory. This one would be different, though. This one, in theory, I mean, what if this really is some kind of a callosase? You wouldn't induce it, presumably, unless you needed it, and then maybe this would help break down the papillae in some way. It's just a theory. Maybe it's nothing. Maybe it's just a coincidence. But it is odd that this group would be so widely distributed, and yet never seem to be induced at the same time as the virulence system. So I mean, you know, presence isn't necessarily evidence of importance, but it's kind of hard to ignore.
Yeah. Okay. I am cruising through YouTube. There's no comments there. If anybody has comments, please tweet them at Microseminar or join into the Hangout here and just ask them. Otherwise, I will just capitalize on all this time and throw other questions at you.
Yeah.
So you think that there's a progression of the type 3 effectors? Because I mean, so the model is that they all get turned on by HARP-L, but it kind of has to be that they're all turned on at different levels at different times, and then there's preference for who gets through the needle. So do you see any of that at all, or?
Oh, in the gene expression? You can see a little bit. Let me screen share again, and maybe we can see if we can interpret that a little bit. One thing that I would say is that there does seem to be some evidence of that. I guess I'm not screen sharing. Hold on a second. Let me do that again. Turn the screen. Come on, come back. You're in tech. Let me go back to the expression data at least. Now, you know, we should always take expression data with like a grain of salt, right? Because we know that it's not necessarily the same thing as protein. So we know that there's a hierarchy of type 3 substrates from work in Xanthomonas, but usually that's thought to be basically between the components of the type 3 filament itself, the translocators, and then the effectors. You would presume that there's also an order to the effectors, and at least when their functions are needed. So for instance, some effectors seem to be really important for this early dampening effect, but others not so much. Others seem to play a contribution in the development of symptoms, which doesn't happen until 2 or 3 days later. So you would think that they wouldn't really be delivered or needed until later in time. I don't know if there's a ton of evidence to support that. I mean, you can definitely see that there's a high level of differential expression happening. You know, some of these effectors that we see as conditional, like for instance, HOP-K, basically isn't turned on at 1 hour at all. So I guess that implies something about an order, perhaps. I'm not sure. I think that in Salmonella, at least, it's been shown that like they're not regulated necessarily at the transcriptional level. Their ordering and their contribution and their importance to disease is actually facilitated by their turnover rates inside the host cell. So it could be something like that. Anyway, did that answer your question?
Yeah, it's good. There's my question.
If I didn't ask the question, I mean, I'll ask the question you asked instead of the question I wish that you'd asked. What? If you mean specific examples, I don't know if we've seen that specifically. I would say, I mean, there are some examples, I suppose, where the expression seems to be pretty differential. But I don't know if that's enough to necessarily say that there's a distinct order yet. So the hypothesis is yes, but the evidence is lacking.
Yeah. Yep. Do you think this will work? Will your transcription stuff work in other plants, or is it like is Arabidopsis like the perfect system, or you think it'll work kind of broadly across? I guess I don't know about.
No, I can tell you for one thing that Arabidopsis is actually probably one of the worst hosts to try to do this in, just because you need so much tissue to get it to work. Actually, it works much better with Indiana and tomato because they have larger leaves, which makes it easier to get a larger amount of bacteria back from this kind of a process. But it does work in Arabidopsis, and Arabidopsis obviously has its value as the model. You know, we can couple what we've seen with plant mutants, and it has the best understood plant immune system. Probably the second runner-up, of course, would be tomato. So we're in a good situation to try to use both.
Cool. All right. No questions there. No questions there. So. I think we'll sign off, and then we'll chat afterwards about some other stuff. But thanks very much, man. That was actually awesome, and it's a good. It's the best demonstration I've seen so far. You can actually do this stuff in plant host with the RNA-seq, which is nice to actually see because it's. Well, I think
it's funny. A lot of stuff has been done on this gene expression, but it's pretty much all been done in vitro with very, very, very few examples. And it's obvious that the in vitro systems really don't recapitulate the entirety of what we know about virulence. For instance, the normal media that we use to induce things doesn't turn on carnitine. Carnitine is one of the strongest things we see turned on by 5 hours, but the media that we use to induce the HARP system, no carnitine expression whatsoever. So yeah.
Cool. All right. Well, I'm going to stop the broadcast. So we'll do